Very well done. So we're looking at carbon and the molecular diversity of life. So we, you and I, and everything we're looking at is that is alive is a carbon-based life form. So we're a carbon-based life form. Indeed, all of the life forms that have died in the past have left us their carbon so that we can use it as fuel. So fossil fuels are the remains of carbon. And it's the backbone of all life in the form of hydrocarbons. What is a hydrocarbon? Can you tell me what a hydrocarbon is?
It's a chain made
It's
of carbons and hydrogen atoms.
Yeah, a chain made of carbon and hydrogen. Yes. So I'd like to talk about the study of carbon compounds. Organic chemistry is what we're delving into now. And I'd like you to keep in mind as we go along why it is that carbon is the molecule or the atom rather of life. So how can so many diverse molecules be formed?
because it can
In
because
many different
carbon
ways.
is
Yeah.
able to bond with itself
Yes.
and create
It can bond with
diverse.
itself.
Yeah.
Yeah. Yeah. And other atoms as well. And in many lengths of hydrocarbon chains with many different kinds of functional groups. So we're looking at those today too. Lots of different forms, isomers. And there are a small number of chemical groups that are key to the functioning of biological molecules. So organic chemistry is the study of carbon compounds. It comes from a kind of a misconception that only carbon-based compounds were connected to life, but of course the enamel of your teeth and minerals and such, they are not all just carbon. There are some non-carbon elements to life forms. Well, we're 70, 95%, plants tend to be around 95% of water, but carbon makes up the rest of the compounds. Organic compounds are the molecules of life. Organic compounds can range from very simple, like carbon dioxide or methane, to complex molecules like proteins. The number is inexhaustible. And while the percentages of elements don't vary too much amongst species, there's variations in organic molecules that can distinguish between individuals of the same species. So living matter is distinguished from non-living matter by such things as and you've mentioned these already today, here and there, carbohydrates, proteins, lipids, and nucleic acids. Carbon atoms bonded to each other and with other elements. So carbon, carbon, but also carbon, hydrogen, carbon, nitrogen, carbon, oxygen, carbon, sulfur, carbon, phosphorus. There are other atoms that can bind to carbon, and between all of those, you come up with an enormous variety of molecules. Carbon won't form ionic bonds. It's not going to just lose its electrons. Its electrons are fairly tightly bound to carbon, but it completes its shell by sharing instead. So it won't lose electrons in its outer shell, but it will complete it, the octet rule, if you recall, by sharing electrons with other atoms. That's known as tetravalence. Tetravalence, which is, let's go up here. tetra, having 4 possible places with which to bond. Organic compounds contain carbon and at least 1 hydrogen. Combinations of carbon and hydrogen form hydrocarbons. Gasoline, propane, methane, ethane. Those are all hydrocarbons. Organic compounds form biological molecules, carbohydrates, lipids, proteins, and nucleic acids. And they're made from simple sugars. So these may be polymers of what are known as monomers. So monomers make polymers. Simple sugars, amino acid nucleotides, and fatty acids, those are monomers. They combine to form polymers. Poly meaning many. Organic chemistry, the study of carbon compounds, not necessarily synthesized by life forms. They may be synthesized by non-life forms as well, which was shown by Stanley Miller in this production of amino acids. He thought that these conditions, the atmosphere containing lots of methane, hydrogen, water vapor, may have been the early atmosphere of the Earth. And so he did an experiment to see, well, can you get organic molecules from these inorganic ones? And sure enough, it turned out that you can. So we did synthesize amino acids using this method. So why is carbon so diverse? The capacity of bonding to 4 other atoms. The key to an atom's chemical characteristics is its electron configuration. Nitrogen has 3, carbon has 4. Well, it's very odd, you know. Oh, this is the, if you're generally an optimistic or a pessimistic kind of person, is carbon's valence shell half full or is it half empty? You know, like the glass. You know, it's weird though. So if you go down the table, the periodic table of elements, what is right underneath carbon? So if you're in a column of the periodic table, what's similar among all of those different elements down one column? What is similar?
The charge?
Not so much the charge, but the number of electrons in the valence shell. So it's the element that's directly under carbon is silicon. Silicon also has 4 electrons in its outer shell. Why isn't it the molecule of life or the element of life? Why doesn't everything just form?
Because silicon
Associations.
likes to bond with oxygen more than it likes to bond with itself.
Huh, okay. I didn't know that. Well,
It's too big.
it is too big, yeah. It's too big as well. So the electrons in that outer shell, well, they're a little more unstable because they're further away from the nucleus, really. Well, I think those are all good reasons. Well, there is a lot of silicon on Earth, and I think there's been movies that have portrayed silicon-based life forms on other planets. So how do they form such diverse molecules? By bonding to 4 other atoms. And that gives it a tetrahedral kind of shape. The bonds, of course, are covalent bonds. They are covalent bonds, not ionic bonds, not hydrogen bonds, not any other kind of bond, but these bonds are covalent. So quite strong. They share electrons. In this case, a carbon is sharing electrons with 2 oxygen atoms. That makes carbon dioxide. Not only sharing 1, but 2. Carbon forming double bonds in this case. 2 electrons are shared between 2 atoms. It forms a relatively flat molecule, a flat and stable molecule, carbon dioxide. Carbon is very versatile. It forms many diverse molecules. Just carbon skeletons, like, for example, ethane, just 2 carbon atoms, ethene, 2 carbon atoms, but some double bonds involved. So there are double bonds, single bonds, and lots of different kinds of elements to which carbon will bond. These are the most common ones. Hydrogen, 1 valence electron to share. Oxygen has 2 valence electrons to share. Nitrogen has 3 valence electrons to share. Carbon has 4. So carbon can form the kind of like a conga line of elements. It's the party animal of the chemical world, carbon. So other sources of diversity for this remarkable element is chain length is almost inexhaustible. Ethane, 2 carbons. Propane, 3. Branching, sometimes they branch. Here's butane, unbranched. Methylpropane is branched. Very similar. It's also called isobutane. So those are fuels that you might use in an industry or to fuel heaters in your home, for example. Forming rings. So often these carbon chains in solution will form a ring that gives them different kinds of properties. Double bonds, forming double bonds. This didn't copy over very well, did it? Anyway, there's a double bond there. And it all depends where the double bond is. It can be within or it can be at the end of the hydrocarbon. So loads of diverse molecules. So they're diverse due to one of the things we mentioned is branching. So here is a fat molecule. A fat molecule. It makes up adipose tissue, and it is unbranched, and therefore its fatty acids are parallel, as opposed to this saturated fatty acid here is straight, but this unsaturated fatty acid has a band to it. And that's because there is here a double bond. And that makes a world of difference in how these molecules behave. Adipose tissue tends to be quite solid. So fats are biological molecules with long hydrocarbon tails. Hydrocarbons themselves are the main components of fuel. From fossilized living forms. Particularly of the large forests that covered much of the earth millions of years ago. What are some other reasons why there's so much diversity with carbon molecules? Well, another reason is isomers. Isomers are molecules with the same molecular formula, but different structures, and as such, different properties. So, for example, butane and isobutane. They have the same molecular formula, but butane is straight and isobutane is branched. So they have different properties, slightly different properties, different melting points, different boiling points, and that kind of thing. So they're known as structural isomers. But that isn't the only kind of isomer that there is. There are also other kinds of isomers known as geometric isomers. So they have the same covalent partnerships, but differ in their arrangement around a double bond. So same molecular formula, but a different arrangement around a carbon double bond, because a carbon double bonded to another carbon, it doesn't rotate, because the double bond keeps that in one plane. So geometric isomers, called cis or trans, depending on which geometric form they take on, they're very instrumental in how cells function and how vision functions, for example. So retinol, for example, retinol has a cis shape and a trans shape. And it changes between these two shapes with energy. So when it changes, when light changes cis retinol to trans retinol, that's a trigger for depolarization of the photoreceptor and thus a nervous signal to the brain. So it's instrumental in vision. Light inducing change in retinal within the rhodopsin protein, which is the pigment that is triggered in vision. So those are structural isomers, geometric isomers, and Enantiomers is another kind of isomer. They're very important in the pharmaceutical industry. And what they are mirror images or mirror arrangements of four different groups. There has to be four different groups or you don't get mirror images. It's kind of like having a left-handed and right-handed version. A left-handed, right-handed version of the molecule. Usually, one form is active and the other form is inactive. So L-DOPA is effective against Parkinson's, but D-DOPA is biologically inactive, same molecular formula and enantiomers of each other. The drug thalidomide, you might have heard of that. Oops. That was prescribed to treat morning sickness in pregnant women, but only one isomer was useful for preventing morning sickness. Unfortunately, the other isomer caused birth defects. So that was a very severe outcome. So one isomer prescribed for morning sickness. The other isomer and some babies were born without limbs. So it's quite serious. So far we focused on carbon skeletons, but what is attached to the carbon skeleton impacts the form and function of these molecules. And there's just a small number of chemical functional groups that are key to the function of biological molecules. Depends on the molecular components attached to the skeleton. Functional groups can replace one of the hydrogens in the four shared electrons. They're the groups that participate in chemical reactions and change the molecular shape. They give organic molecules very distinctive chemical properties and interact with different targets in the body. If we're talking about humans, we've got estriadol, which is synthesized from cholesterol, and testosterone, which is synthesized from cholesterol. What are the differences? Well, some of the differences are the location of the methyl groups, and another difference is the location of double bonds. But it is different enough that they have vastly different effects because they bind to different receptors. Due to their structural differences. Of functional groups. So it really makes a lot of difference. What are those functional groups? There's 6 that are important to the chemistry of life: hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, and phosphate. So those are the ones that we're going to look at. Let's take a look at hydroxyl. There it is there. It's just simply an OH group. Sometimes it's written HO. It's a hydrogen atom bonded to an oxygen atom. The oxygen atom is bonded to the carbon skeleton of the organic molecule. It's not the same as free OH. I'm just going to go down to the next one before we go over to the right because they're underneath. These are known as alcohols. Yeah, they usually end in "-ol", like ethanol, for example. So here's ethanol, and there is the OH. So what is the consequence of that? It's polar. It's a result of the electronegative oxygen atom drawing the electron towards itself. It attracts water molecules, and very importantly, it helps to dissolve organic compounds, such as sugar. So a lot of sugars have hydroxyl groups attached to them, and they're easily dissolved. So that, my friends, is the hydroxyl group. The carbonyl group. Your version of this lecture, by the way, that's online is nicer. In the process of copying it over, I guess, from my computer to my iPad, it lost a few things in the copying. Don't know why. Anyhow, carbonyl group, CO, is a carbon atom joined to an oxygen atom by a double bond. It's a ketone if the carbonyl group is within a carbon skeleton, such as here. This is acetone, very simple ketone. It's an aldehyde if the carbonyl group is at the end of the skeleton. It's here, it's at the end, in which case it's known as an aldehyde, like propanol. Aldehydes are easily oxidized. And ketones are not. So they may be structural isomers, but with different properties, a ketone and an aldehyde. So cinnamon, for example, cinnamon as an aldehyde has a very sweet smell. It's very volatile. Carboxyl. Our carboxyl group has carbon, 2 oxygen atoms, and a hydrogen atom. 1 oxygen is double bonded to the carbon, and the other OH group is a single bond to the carbon. That's a carboxyl group. They're also known as carboxyl acids or organic acids because they readily give up this hydrogen. This particular one is acetic acid. That gives vinegar a sour taste. Yeah, it has acidic properties because it is a source of hydrogen ions. The covalent bond between oxygen and hydrogen is so polar that hydrogen ions tend to dissociate reversibly. In cells, they're found in the ionic form, which is called a carboxylate group. All right, let's look at some more. Amino, that shouldn't be there, should be over here. Phosphate. So our amino group is a base. It has a nitrogen at the end with 2 hydrogen atoms attached to the carbon skeleton. Amino acids have a carboxyl end to them, but also an amino end. That's why they're called amino acids. So glycine is both an amine and a carboxylic acid. And those are called amino acids. So the base, the amine, acts as a base because it can pick up a proton from the surrounding solution. In this case, we have ammonia, and it picks up a hydrogen from the surrounding solution. Sulfhydryl, another functional group attached to carbon skeletons. It consists of a sulfur atom bonded to an atom of hydrogen. It resembles hydroxyl in shape. Ethanethyl has a sulfhydryl group attached to it. But what's very handy is that two sulfhydryl groups interact to stabilize protein structure. So, for example, the protein that your hair is made of, keratin, there's lots of sulfhydryl, they're called disulfide bonds when the two sulfhydryl groups make a disulfide bond. And they're very strong. But you probably weren't around in the 80s, but definitely not. But the 80s were a time of perms. So you got like your hair, if you had straight hair, everyone wanted to have curly hair, and if you had curly hair, you wanted to have straight hair. And so you go and get a perm, which meant that you could change your hair configuration. And that's because the chemicals will break the disulfide bonds in the proteins in your hair. Then the hairdresser curls your hair into a curl and then adds another chemical later on to form new disulfide bonds, and that retain that shape for a little while anyway. All right, here's a test. Can you identify a common functional group in these molecules? And you're going to say, are they the ones that are highlighted? Yes, they are. Yes, hydroxyl groups. There's lots of them there. How about this one? There are three different groups. Do you recognize them? An amino group, yes. An amine group, good. Hydroxyl, yes. Excellent. And one more. Phosphate, good. And this, of course, is ATP. This is the molecule that provides us and most other living forms with energy. It's an amazing molecule. Now, one thing you'll notice is that, oh, negative, negative, oxygen, negative. Well, as you know, negative ions repel each other. You're like, why are these attached if they're all negative? Well, they are loosely attached. This last phosphate group is very easily removed. And that is what makes ATP such an effective source of energy, because that phosphate group, it just leaves quite readily and joins other molecules, changing their shape, changing channels in cell membranes. It's very, very effective, ATP. Can you see any here? What are some common functional groups here? Carbonyl groups. Good. And here's a methyl group and a phosphate. And here, There isn't a phosphate on this one. No, but the other one, yes. But what about this one? A carboxyl group, good. Methyl, and what's this one? An amine group, good. Excellent. All right, thank you for watching.